Hello everyone, my name is Alda Cahyaningtyas and this is a video for microbiology. It's the lactic acid fermentation lab setup. I have here with me green cabbage, one head of green cabbage that I have washed, and I have also removed the bruised and soiled leaves from the cabbage. So it leaves us with just almost perfect cabbage without any dirt on it. And what I'm gonna do next is I'm going to cut this in quarters, and then I'm going to chop it finely. And it's also important for you to make sure that the size of each chopped cabbage is kind of uniform all throughout. I have just chopped the green cabbage. I forgot to mention earlier that before you chop the cabbage, after you cut it into quarters, make sure that you remove the pit, and then you can proceed with chopping or using food processor to chop the green cabbage uniformly. And here I have chopped, I believe this is kind of too much for the experiment, so I'm gonna get my measuring cup out and I'm gonna move that to a separate bowl. We're gonna need 4 cups of packed green cabbage into the other bowl. So as you can see here, I have the cup, the 1 cup measuring cup, and then I'm just gonna scoop it right like that and then pack it. And then I'm gonna pour this into another bowl over here. Actually, we might have just enough. Not too much, not too little. Okay. So with this 4 cups of green cabbage, I'm going to add 4 teaspoons of salt into it. This is my salt, this is my teaspoon. 1, 2, 3, 4. And then we're gonna mix it really well. After mixing it really well, now we can leave it for 2 hours, or at least until there is some liquid that can be pulled out for our initial pH test. I have left the cabbage for about 2 hours, a little over 2 hours. Now I'm going to set aside 1 tablespoon of the juice that is in here right now, and then I'm gonna put it in this baggie. This is about 1 tablespoon of the juice, and then the rest over here we're gonna transfer it to this quart jar. We're gonna fill it up about 2 thirds of the quart jar, and then I'm gonna probably use this to compress the inside, the content of the quart jar. I'm gonna fill it with water and then try to fit it inside of the quart jar. Otherwise, if this doesn't fit or just like doesn't provide like equal pressure to all the surface, I'm gonna use just like a sandwich bag to help me compress the cabbage. And I'm just gonna try to make sure that it's back without crushing the cabbage. This is done. We're gonna place not this one. We're gonna pour some water in this bag, and then we're gonna put it in here. Let's see. I have placed a Ziploc bag with water in it to add pressure to the cabbage, make like a compression to the cabbage, and then I've placed the lid on top of it. To maximize the seal, I'm gonna put tape around the lid just because I feel like this is still easily openable, or accessible. I'm gonna seal it, and then we're gonna leave it to ferment for 11 days.